Good morning, everybody. First of all, I would like to say many thanks for organizing committee and especially Professor Dr. Hala Gab, Dr. Wael Abul Khair for inviting me and give me the opportunity to present my work that was done recently in Upper Egypt in our Tissue Culture Molecular Biology Center. My talk today about the cancer stem cell again, but I mainly focused upon the cancer breast stem cell. So, my talk about the cancer stem cell related gene, exactly periostine, AGPL2, as a novel prognostic markers for breast cancer. My agenda today, I will discuss the background and what about the normal stem cell and cancer stem cell and difference between both, characters of cancer stem cell, and about the origin of cancer stem cell, and about brief also highlight about the differences in the markers between different tissues as regard the stem cell or cancer stem cell. Then we will discuss what about our aim of the work in this research project, material and methods, our results, and conclusion finally, and recommendations. Background, what about normal stem cell? As you know, it's a rare cell within organ with the ability to self-renew, give rise to all type of cell within the organ just to drive the organogenesis. But cancer stem cell, again, it's rare cells also within the tumor with the ability of self-renewal and giving rise to the phenotypically diverse tumor cell population. So it's capable to drive tumorigenesis. The characters of cancer stem cell, it's considered a distinct population with multiple unique features to cause them vital for tumor formation, as I said, tumorigenesis, and the capability of self-renewal, so they are immortal, what we call immortal cells, necessary to maintain the population of tumor cells. Again, it has a pluripotent capability. That means it can generate a lot of tumor cells with different phenotypes. What about the, actually, cancer stem cell accused that they are responsible mainly for the recurrence of the tumor, for the metastasis, either nodal or distant metastasis. Again, several problems as regard the cancer patient, as regard the chemo resistance, radio resistance of this patient, and as I said, a lot of metastasis, they are accused that they are responsible for all these problems. That's why it's a matter of hot interest recently. Ongoing research projects, several ongoing research projects have focused upon these cells, hopefully to make a sort of eradication of cancer stem cell from the tumor, so we can make what we call personalized cancer medicine. So they are considered a migratory cancer stem cell, self-renewal, differentiation, chemo and radio resistant, I said, invasive and metastatic. First, let us say, what about the origin of cancer stem cell? Cancer stem cell has one of two main origins. There are several theories. Maybe a transformed adult stem cell, so it may be stem cell as usual, but transformed, some sort of genetic transformation of these cells, or genomic instability of cancer cell itself. So the cancer cell itself, it's not the main. Sometimes the biopsy indicating that several morphological criteria or pathological criteria of tumor cells, but not all these cells are responsible for the metastasis or recurrence or. So that's why the genomic instability of cancer cell may be the origin of cancer stem cell. As regards the different types of cancer, what about the cancer stem cell? Is it the same for markers in different types of tissue? The answer is no. Actually, different examples of cancer as regard acute myeloid leukemia, breast cancer, colon cancer, gliomas, nasopharyngeal carcinoma, and so on. So a panel of markers completely different. My focus today is about the cancer breast. So CD44 positive, CD24 negative cells. So when I try to make sorting for these cells, actually we have to focus upon the group of cells that have the 44 positive, 24 negative cells. Historical view about the identification of various cancer stem cell. Actually, it was 1991, and it was in the cases of acute myeloid leukemia, and the origin of the cell responsible for cancer stem cell formation, it was the hematopoietic stem cell. This is the historical issues. So several authors tried previously to characterize cancer stem cell and at single level, and to understand the genetic and biochemical mechanisms, the properties of both normal and cancer stem cell, and also others try to characterize the response of cancer stem cell to chemotherapeutic. Others try to develop a therapeutic strategies. This is the era of hot research or ongoing research now, to develop the therapeutic strategies to target the cancer stem cell to prevent tumor or cancer recurrence. Also, accumulated evidence established that cancer stem cell population are more resistant to conventional therapy than non-cancer stem cell population. Therefore, it's a must, it's a crucial, very important to eliminate these cells to treat the malignancy. Also, several, as I said, several therapies targeting these cells and multiple novel try to change based upon changing the microenvironment, the niches. So the trials were focused upon the altering the microenvironment or niches for that support the cancer stem cell. Others try several strategies, different strategies focused upon the cell itself, while still in preclinical and clinical evaluation as a promising new therapeutic strategies. What about our aim of work? Our aim of work in this study, there's some controversy regarding both periostine expression, nature of periostine producing cells within the tumor. So we used quantitative RT-PCR to assess periostine expression in breast cancer tissue, and in parallel, we make some sort of characterization using CD44 and 24, which is well-known marker for cancer breast stem cell. From clinical specimen already used flow cytometry, as I said, and the RT-PCR, correlation analysis between the percentage in all tumor samples. I want to focus upon the all tumor sample. So in this study, actually, we based upon the all cancer biopsy that was taken from the cancer patient. We didn't make sorting in this issue. In this respect, we are focusing upon, is it already have upregulation or increased expression of periostine and the AGPL2 as a nearly known marker, predicting or helping in determination of the presence of highly virulent cancer stem cell. So we try to make this, but without sorting of this cell, we make it on the whole tumor tissue and try to make this correlation with several markers, as I said, first with periostine and the AGPL, hormonal markers, HER2/neu, ER, estrogen receptor, progesterone receptor, and histological staging, what we call histopathological staging, lymph node metastasis, and clinical staging. So we try to identify, is cancer stem cell, make a correlation, the percentage of this cell with the periostine, AGPL2 expression status, and to evaluate the clinical implication. The gaining this knowledge will lay foundation, actually, for managing breast cancer using personalized cancer medicine. So our design was total number of 100 patients, histologically confirmed cancer breast, who underwent a radical operation in the Asiyut University Hospital between January 2016 and January 2018, enrolled for immunohistochemical. Okay, qPCR flow, mean age of patient was from 27 to 80, and again another 100 of patient with benign tumor as other arm of our study, and these patients equally divided, as I said, to two groups. Each group was directed for sorting and by flow, and also for qPCR and for hormonal or histopathological. The criteria, curative operation will be were made, resected biopsy pathologically examined, more than 10 lymph nodes pathologically examined also, complete medical record, ER, BR, here to new, and so on, and ethical committee approval, first of all, ethical committee approval from our Faculty of Medicine at Asiyut University, and also written informed consent from all these patients.